works.
Excellent. Thank you again. In the interest of time, since we have about 12 minutes left, I have put the Jeremy and the team on extremely short notice. I think I approached them probably 20 minutes before this call just to see whether they wanted to give a brief update on the preprint that they put out. I think it's a fascinating look at convergent evolution and some of the things that we're starting to uncover. It's also a great story of cross spheres collaboration. So with that, normally I would give an update on some of the national surveillance efforts and where we are in sequence data. I'll probably kick that over until next week, but what I will do is post the slides that have at least updates of what's in GISAID and probably some of the other updates as well. Jeremy, I don't know who's going to drive, but I'll switch over to you.
Yeah, I'm open to Daryl or Emma taking over if that ever becomes helpful. I don't know if I'm allowed to share my screen yet.
Yep, you should be good. Sorry.
I have. Yeah, sorry. Can people see my PowerPoint?
Yep. And if this is what you're able to put together in like 20 minutes, I am seriously.
It's actually just it's our slide deck from preparing the manuscript. There was zero sphere specific preparation on 12 minutes notice. But so yeah, this came together really both spheres, but also of course essential role for GISAID, because we wouldn't have known about Daryl's data, Daryl, D&D as they call themselves, I think, in New Mexico. And they wouldn't have seen ours if everyone wasn't uploading their data rapidly and promptly. It was amazing to have just Emma's fluency with next strain auspice auger at her fingertips, and of course, Vaughn Cooper's expertise in just studying how microbes evolve. Like, he's a microbial evolutionary geneticist. So as a virologist, I tend to be very cautious in a pandemic about saying that a mutation at a site might be giving an advantage or even a signature thereof, because so many mutations can be fixed purely by chance. So we saw at our local surveillance efforts here in Shreveport, Louisiana, where we do a lot of congregate facility testing. And that's another reason I was kind of like, well, okay, it's 20% of our cases from December, but so many of these are congregate facilities. Do we really have any signal here that could be meaningful? And the thing that set me off was, well, this is right near the furin cleavage site. It's not really a furin cleavage site. There's 3 different proteases that can cut there. Tempress-2 is more of a trypsin-like protease. And then in the context of pathogenesis, as I've learned from my collaborator, Benhur, there's also neutrophils will spit out things like elastases. So all those things can be involved in the complex biology of coronavirus entry. And so there's basically 2, if you listen to people like Jason McClellan and Barney Graham, Cosmetia Corbett, people who study coronavirus entry professionally, there's more than one protease event that are critical for entry. In fact, the furin cleavage site, the polybasic site that this is next to, is really important for pathogenesis, but it's not even absolutely conserved in coronaviruses, and it's not essential for fusion. So it's important to know those kind of like background things. And I also want to give a shout out to Keith Gagnon from Southern Illinois University, because I wasn't fully, I saw some stuff in the news about an Ohio variant, and I heard of some of his research, but in the rush to put this together, we failed miserably to cite his paper. And he and his group very nicely showed that there was a one of the lineages that I think we named Robin 1, or Robin 1 and Robin 2, what it was something that his group had already published on probably a month before us. So they specifically called out the 677H mutant. So as anything in biology, it's very common that other groups, and you're not the only one figuring stuff out. We owe that group credit, and we will certainly cite them before we go into a peer-reviewed publication. So anyway, this is a chart made by using GISAID data and our data analytics team using Tableau here in B, and it's just showing the incidence of these polymorphisms affecting position 677. And then Vaughn helped make this graph on the left, just showing the incidence in the world and in the United States of viruses carrying this polymorphism. And it's kind of striking just because we're so many months into the pandemic, yet the emergence of variants affecting this position seems to have been delayed. And why would that be? So one thing that's, I think, notable is, we learn in 10th grade biology class that certain types of mutations are more common than others. And I'm sure there's levels of chemistry that are complicated here because you have interferon regulated genes that modify certain bases, like adenosine deaminases, cytosine deaminases, and those, or cytidine deaminases that can add some complexity here to the spontaneity, if you will. But we do know that transitions are more common than transversions all over biology. And it's one notable thing is that any of these changes here involve transversions. So either a purine base going to a pyrimidine or vice versa. So those are more rare, they're less common. And if I'm sure a lot of people on this Zoom have done analyses of these sort of things, there's already a lot of stuff in the literature. The most common base change that's dominating this genome as it's spilled over into humans is cytosine to uracil. So, and that's catalyzed by apoBACs. So that could be a signal of host defense ISGs that are upregulated that are trying to hyper mutate the viral genome to protect the eukaryotic host. I don't think that's really been proven yet, and it's surprisingly rare to find proof for that for an RNA virus. But anyway, so this is the tree that that Emma made, and when we started freaking out a little bit more than just, at first I sent an email to Vaughn saying, hey, we're seeing a lot of this 677P in our data. And I showed a little tree with there was a little bit of H in there because I was just looking at Louisiana. And he did a more thorough analysis, and we thought we had something maybe. And then when Emma noticed that you had examples of things that looked like one clade or one sublineage, but in fact there were two different viruses in there, because tucked under the hood were either a change to a U or to a C. So you have the virus coming up with both different histidine codons, which is kind of neat because it's flipping from obviously a glutamine to one of two different histidines. And we're seeing it at least 6 different nodes for, six different sublineages for histidine. And we have only one so far to date sublineage is a B.1.2 or next chain 20G, that is we call it Pelican, that's changing it to a proline codon. And my hunch is that this proline codon change is probably, actually, if you look at the data, it is one of the least common types of change from an A to a C. So it's probably harder for the virus to explore that sequence space spontaneously. And it's not in our paper, but there's also a cluster of arginine mutants. It's just much smaller, and we don't know if there's more clusters of them out there, and they're just undersampled, because there could be a robust lineage of 677 arginine mutants out there. And I also have to, I think, give credit to Keith Gagnon there, because I think he had even in his preprint a little sliver of R as well. So by no means are we the first to point to these polymorphisms. I think the thing that's novel about our work is through serendipity and collaboration, we noticed that there were multiple lineages of this doing this independently, which is obviously a signature, as Vaughn Cooper would tell us, of parallelism. And in an interview with a reporter from Science Magazine yesterday, he said something quite memorable, which is when you see parallelism in a well-fashioned in vitro or real world evolution experiment, you can take that to the bank, that it usually means something real. So that's what the cartoon looks like one slide earlier. But if you wanted to look at, and Emma, if you want to take over at any point with your actual build and click people through, by all means, because this is 100% Emma and Daryl Domman who came up with this figure together. So I don't want to take their thunder in case they'd like to take over the presenting role. Anyone? I'll pause for 15 seconds, see if there's takers.
I'm happy to walk through the next strain tree if people are interested, but you've definitely covered the main points, so it just depends if it kind of adds, but yeah.
I think people might get a kick out of it because you're much more fluent, and it was really a discovery that there were multiple sublineages here. And it's really cool seeing her click through, and you see some of the clusters on her build change color if you're looking from amino acid to the base. And I think she could show you exactly what I mean. It's really quite cool. So I'll stop sharing and let her show you guys if people are interested. I don't know how much time we have.
I'll try and give a very brief. So so hopefully you can see my screen now. So when we started looking into this, of course, I just went and made it what we call focal builds. So this means I took all of the sequences that have a mutation at position 677, and I threw those into our kind of mixing bowl. They select out any sequences that are relatively nearby and then some background. So keep that in mind as you look at this tree. It has been selected to be enriched for 677. So it is not a proportional representation of the number of mutations we see. But I think the first thing that kind of grabs your eye, so I've colored this tree by 677, the kind of ancestral state is Q. And you can see that there are a number of different amino acids, and they're really across the tree and across time. So this is something that has popped up quite a number of times in the past, and we see it, around the world. But I think what's really noticeable is that already you can start to see that there are some really noticeable clusters here. So of course, here's the pelican. It stands out because it's the only one with P. That's globally, it seems to be the only one with P. But then we can even see within H that there are some really noticeable kind of clusters here. So if I filter down so that we just look at HNP, sorry, my computer doesn't like to do zoom and next strain, but we'll get there. And then just filter to the USA, you can start to see what we started seeing with these kind of independent arising. So you can kind of start to see that there are some groups here. And if you switch to divergence view, it becomes a little bit more apparent that these are kind of different. For the robins, you have to kind of go in there and zoom around and look at some background to figure out that these are independent arisings. But actually, if you look at these in the North American build, there are other sequences that go between them that don't have the 677H mutation. And so we're pretty confident these are also separate. Just to make that a little bit clearer, we can color by the birds. And go back to time view if it thinks it can. And so that's kind of the prettiest picture of these lineages, and you can kind of see where they're found across the US. There is some kind of interesting distributions here in that, some are definitely more common in some areas of the US, some are more common in other areas of the US, but they're mostly found fairly widespread. I think that bluebird is the only one that is really, really stuck to the Northeast. The other ones are found across the US. One thing I just want to finish on really quickly is kind of, here's one I prepared earlier. This is the same tree. Come on, computer, you can do it. The same tree, but this time I haven't filtered to the US. So this is showing a bit more globally. And I've just picked out a few clusters that are international. So for example, this is in Denmark. There's a couple in the UK. There's a couple in Australia. This one is actually within V1. It looks like 677H has popped up a few times between, sorry, that's B117 for those who use the Hangul lineages, the variant predominantly found in the UK. So I just wanted to really emphasize that we do see this more than in the US. We just really focused on the US for this manuscript, but it is something that we see recent clusters even outside the US. So yeah, whistle stop tour.
Wow. Yeah, so you can see why we were stoked to have Emma on the team. She really drove the parallelism argument to the first observing it and then bringing it to the whole next level. So yeah, I mean, I feel like when I was a 7 or 8 year old, I went to like fishing camp with the elementary school, and my fishing line got tangled in a tree, and I was the only one who caught a fish because a giant trout jumped up and bit the hook. I found this random variant and sent an email to the right person, and she turned it into a Mozart symphony. So and then so the other people who brought magic to this story, I'm going to skip this slide. Let me go back to sharing screen, is the structural biologists and the cell biologists who study entry. Because let's share this. I think we can skip this or just go over it quickly. But this is just a little cartoon showing that these are different lineages coming up with the same mutation. And as a stand-in for the experiments we'd like to do, we have a cryo-EM structure, and this is one with all of the RBDs down. But as we know, like these coronavirus spike proteins, they kind of unfold in a multiple step process. It's really Jeremy Lubon's lab, together with, of course, Bet Korber and I can't pronounce the name. The person who showed first that S1 subunit shedding was sorry, I'm getting like by on 2 hours sleep lately, but there was a really powerful paper that came out in the summer that showed from Scripps research team in Florida. I can't remember the person's first name. I suck. Anyway, her group showed that S1 subunit shedding was faster in the D614G or Doug lineage. And that all goes back to differences around this area where the polybasic site is, and 614 is a little bit away, but it makes it more flexible. So probably the spike can breathe a little more, and then it starts to interact. You know, it has 3 identical subunits, and they start to breathe and interact with ACE2 molecules. And when the trimming occurs in this polybasic region, the polybasic region is found on a disordered loop if you talk to the cryo-EM wizards. And apparently if you have a histidine here or a proline here, you have different mechanisms that might make it a little juicier for a protease or a more conformationally labile, so that the end result is you have a more metastable fusion protein that's able to more dynamically interact perhaps with and change the spectrum of different protease activities that might unlock it. So you have a real footing in biochemical mechanisms that might expand the tropism of this virus, in particular for, as Stephen Goldstein told us, you know, the bat viruses that these probably came from are GI viruses, and the GI tract has different protease activities associated with it than the respiratory tract. So this could be an adaptation to different host tissues or other things. And I think we should also point out that the POO mutation, POOH or 681 of the B.1.1.7 variant, you have a histidine mutation at the very first proline of the actual polybasic site. So there's strong precedence, and there's a signal from maybe evolution here that substitutions around the furin cleavage site, or we shouldn't call it furin cleavage site, it's a polybasic site, could be influencing tropism and pathogenesis, or maybe host adaptation. But I think that's enough. And thank you all for your attention.
That was wonderful, guys. And I'm sure it will spur some additional conversation in and around that. It also reminds me that the nomenclature debate rages on. I think that the addition of bird names is a lovely addition. I especially like the regional addition, pelicans, for some of the variants that were identified. Okay, never mind. I don't know if that was, I won't get out the Audubon book, but yeah.
No, I put that in the manuscript, and Emma reminded me that we kind of want to get away from regional, but we thought that it would be kind of a between the lines. Well, the pelican, Louisiana, is kind of a dead giveaway, isn't it? But yeah.
For the other birds, there are definitely kind of regional associations, but they aren't linked to one state. And so I think that, and also we have like mixed up some of the names because we liked others better. So there's a loose connection, but you can't really trace it back, which I think is ideal.
No, it's fantastic work. Any comments? I know we're over time already, but I think thank you so much for putting this together last minute. I think it tells a fascinating story, and I think more of these connections will start to be drawn as we start generating more data and have more data to share.
Hi, I'm Venkata from Maryland. I have a question, like considering the potential of these S667 and 677 variants, and also another one that was discussed in the paper that is a lineage B.1.525, which has the mutations of both the UK variant and also the African variant. Are we considering them as variants of concern, or at least under investigation, or we are waiting till their numbers increase more?
Yeah, well, I think when reporters ask that question, we always try to say the same thing that we probably say here. In the case of, if it's B.1.1.7 that has this on top of it, I'd say you're already in a variant of concern, and the onus is upon you to prove that this change made it less of a variant of concern. Any other variant that comes up with a histidine or a proline at 677, there's no empirical proof yet that these have anything different than, you know, 2 marathon runners going by your house, one running a mile 1 second faster than the other. We have no reason to believe that this is anything other than a change that might give the virus benefit after getting through 30 different people in a transmission chain, i.e. winning the evolutionary race, but not being any different as far as a measurable effect in a short period of time. So I think we just have to be very cautious, especially when we talk to journalists, but even among each other, until there's proof, it's not really a variant of concern, it's just a variant. And there's so many variants.
Well, yeah, you know, I think that is a fair comment because, you know, some of the headlines definitely read 7 new variants identified across. I mean, that kind of became the story, which, you know, I think it's important to have that language.
Yeah,
and, you know, this
thumbs down
reminds
on
me
that.
too, that we need to be, you know, we have some standardized variants of concern, variants of interest criteria that we're working on that's within CDC and across the interagency. I think we'll probably want to get that out sooner rather than later so that people have objective criteria to benchmark some of these variants against, or at least, yeah, I think it's something that would be good for comment as well.
So Duncan,
final
are there
comments?
any specific criteria to judge something as variants of concern? Is there any source? Can you?
We have a working document that we were using internally. It's aligned with a lot of the international community as well. I mean, you know, for example, Andy Rembo just put out a few notices about A23.1 and B.1.525 as 2 things that they're keeping tabs on based on surveillance. You know, again, those are variants of interest probably at this point, but they have properties that definitely make them worthwhile. I think, you know, some of the variants of concern language is complicated because, you know, you look at things like B117, especially as they acquire additional mutations that are potentially going to affect their phenotype or their behavior, like E484K. Whether or not that it fits under traditional language or whether that actually needs to be separated out remains to be seen. But yeah, nomenclature continues to be a challenge, and I think we need to try and nail it down sooner rather than later. But I love the addition of birds. No, Jared, not bird flu. Do not do that.
Thank you.
All right.
That's so funny. Someone said if we need bird watchers. So yeah, genetic epidemiologists can now be called bird watchers. This is awesome. Thank you, Lee Katz. That was really cute.
I do like that. All right. Thank you all. Thank you, Jeremy. Thank you, Emma. Thank you, Daryl. Thank you, Noah. Everyone else that I probably missed, Keith. And we will see you same time next week.
Okay. Yep.
Bye, everyone.
Thank you.
Thanks. Bye.